Today we will start with the advanced enzymology, introductory parts of that. That will include what is enzyme catalysis, how it works, what are the theory behind that, and what are the factors involved for several steps for enzyme substrate binding. So before that, you have to understand what is enzyme catalysis. As I said previously that it is the fundamental process that involved in the biochemistry and pivotal role in every living organism. Every reaction can only be possible with with the involvement of the enzymes. So it has a role of functioning in the living system. So enzyme are a biological catalyst. As we say that it facilitates the chemical reaction. But how? By lowering the activation energy. By lowering the activation energy, it means required for the reaction to occur that lower the activation energy, delta G0. These allow reaction to proceed at faster rate. If you put a biocatalyst, the reaction will enhance the rate of reactions and it will not injure to the reactions or harm to the reaction or harm in the living reaction. Under mild condition it is work. Now making this essential for efficient functioning of any metabolic pathways, you require enzymes. So the theory of enzyme catalysis, remember this word. The theory of enzyme catalysis is based on several key principles. In introductory para or primary language, or you can say in primary or principle of enzymes, you can include the two things, the concept involved in that. First, one of the central ideas, it was proposed in 1894 by the Emil Fischer. What they said, they give the example of lock and key model thesis. This central idea is the lock and key model, and it was proposed by Emil Fischer. And according to these models, the enzyme have a specific three-dimensional structure. Remember that words, enzyme having a three-dimensional structures, and that complement the shape of, as I said previously, lock and key model, the shape of their substrate molecules. So substrate, the enzyme and substrate, it is binding at specific site, and that site called as active site. Active site is a, what is active site? Which is a small region with its structure bind to the substrate molecules, and it facilitates the conversion of substrate into a product. This model suggested that enzyme substrate binding is highly specific, similar to how the key fits into a lock. So we can say that every lock has a specific key. There is no master key in this world, according to the living system. However, subsequent studies and many researchers has that said that not only a lock and key models or operant concept, one gene, one enzyme. Likewise, lock and key model alone cannot fully explain all aspects of enzyme catalysis. So to understand the enzyme catalysis, you should know the knowledge of models of lock and key model by Emil Fischer. Now they have another scientist name, that is Daniel Kostland. He has proposed an alternate theory, and that theory known as induced fit model. This model suggested that the binding of substrate to an enzyme induces conformational changes in both, not only in the enzyme but also in substrate also, that leading to a optimum alignment of reacting groups. Optimal alignment of reactive groups, these words you should try, and thereby it facilitates the catalytic reactions. The induced fit model emphasizes the dynamic nature of enzyme substrate interaction and highlight the importance of flexibility in the enzyme structure. How the flexible enzyme structures and how it interact with the enzyme and substrate, and what are the conformational changes at the reactive site or active site, both in enzyme as well as in substrate. So enzyme catalysis involves several steps, not only enzyme and substrate, including enzyme substrate bindings, catalysis itself, product release, how much product is released, and the rate at which these steps occur determine the overall rate of the enzymatic reactions. The catalytic activity of the enzyme can be influenced by various factors. The factor affecting enzyme activity or the rate of reactions or catalytic activity affected by temperature, pH, substrate binding, substrate concentration, presence of cofactors, presence of inhibitors. This is the concept of or principle of enzyme catalysis. So, if it is asked in the examination, you have to write only these slides.